The overall goal of this procedure is to isolate a population of cell-type-specific protoplasts from a leaf. This is accomplished by first growing plants that express GFP in specific cell types in the leaf. The second step is to harvest leaves and generate protoplasts using an enzymatic digestion. Next, the protoplasts are filtered and sorted using fluorescence-activated cell sorting, or FACS. The final step is to confirm the accuracy of the sorting using fluorescence microscopy. Ultimately, the sorted protoplasts can be used for analysis of cell-type-specific gene expression, proteomic analysis, or metabolic profiling.
Fluorescence-activated cell sorting has previously been used successfully for roots, but its use for isolating leaf cells labelled with GFP prior to this method has been limited due to the interference from chlorophyll fluorescence.
Visual demonstration of this method is critical, as rapid but careful sample preparation is crucial for minimizing the extensive physiological changes that can occur during cell sorting. Generally, individuals new to this method will struggle because they will not be aware of how to set up the cell sorter or how to confirm accurate sorting.
This method can help answer key questions in the plant systems biology field. The problem with identifying gene regulatory networks in a complex multicellular organ such as a leaf is that different cell types express different groups of genes. This genetic information is lost in the harvesting and analysis of a whole leaf.